Next up, we have Kayla Yearout. Kayla, are you here?
Hello.
There you are. Okay.
This beautiful plant known as Brassica napus or canola is grown for the oilseed content. Canola is mainly used for the production of cooking oils and is the healthiest vegetable oil on the market. It can also be used as livestock feed and biofuels. And just like any other crop, pests and diseases affect it every growing season, with one of the most devastating being blackleg. Blackleg is caused by a fungal pathogen known as Leptospira maculans and is a major constraint to the production of canola worldwide. It has the ability to infect the plant during all growth stages, causing stem cankers that can lead to extreme crop damage and yield loss. Although blackleg has been described since the 1700s, it was only just identified in northern Idaho in 2009, making it a new disease to the region and an emerging problem to certified seed production for the Idaho oilseed industry. Other areas around the world have been able to identify the spread of leptospira and how it interacts with its environment, otherwise known as the epidemiology of the disease. But because blackleg is new to northern Idaho, not much is known regarding how our environment contributes to the development and severity of it. Therefore, my research looks at the biology of the pathogen and the epidemiology of the disease. One of the most effective forms of disease resistance in plants is through a mechanism known as qualitative resistance. This is a gene for gene interaction in which a disease inducing gene in the pathogen triggers a resistance response from the plant. Leptospira isolates collected from northern Idaho have been characterized to determine the frequency of these disease-inducing genes present in our region's pathogen population. And it's by knowing these frequencies, we can then determine which resistance genes in canola will provide optimal control against blackleg specific to northern Idaho. Along with genetic resistance, foliar fungicide applications are an effective means of blackleg control. However, it's important to know when these treatments will in fact be the most effective. The time of the growing season when blackleg inducing spores are produced does vary throughout the world due to environmental conditions. It's through the use of spore traps that we can identify when these spores are being produced and causing initial infection, along with the weather conditions that are optimal for the disease onset we see. By understanding when these spores are produced and released, and when they're causing initial infection, we can then determine the optimal time to apply fungicides. In the end, my research will contribute to developing grower guidelines for the best management practices against blackleg in canola specific to northern Idaho. Thank you.
Thank you. Interesting that it only appeared in North Idaho in 2009. Do you have any idea on how it got here? Did it just as a spore blow in with the wind?
Yeah, we think there's a potential of the windblown spores, but also the potential of infected seed lots being imported into the state and then, producing rain splash and windborne spores that then infects surrounding plants and crops.
Are there any places that are that haven't had this disease yet? Canada,
Um,
place up north?
Yeah, so it's all over Australia, Canada, and Europe. However, it's just now being noticed in China. So that's the last country that hasn't been affected.
Okay, and so your work with producers obviously would be important.
Mhm.
How do you see that happening?
So I've already been able to interact with some of the producers, which is an amazing experience. We hold field day tours every season to portray the variety trials and other extension work being done at the university. And I was able to interact with the producers there and tell them about my research and how it will benefit them in the future.
Super, very good. Thank you.
Welcome, thanks.